So I'm going to talk to you about my PhD project, which is the decoding of tactile afferents that are responsible for sensory motor control during object manipulation. So an overview, just give you an overview. So I'll just go through an introduction of what I'm doing, what the objectives that I want to achieve during this project, and a bit of background on the physiology of touch receptors and the methods we're using to build models that are going to give us some understanding of how we can use this information for better understanding what is happening, and then some results that I have, and then a summary and some work that still has to be done, and then we'll be open for questions and comments. Humans have an amazing ability when it comes to manipulating objects. For example, we can easily pick up objects that are delicate without crushing them, and we hold them perfectly fine. And this is to the envy of many individuals that are trying to design robotic devices that can emulate this capability. For example, there was a study that showed that proper grip of objects is adapted to frictional conditions, where they were changing frictional conditions of the surface between the fingertips and the object, and there were forces automatically adjusted without even individuals being aware of what happened. And that sparked a race. A number of engineers came up with try to use this similar strategy in order to design robotic devices, but they use an approach where they were basically trying to adapt to friction as well, but they were using sort of a strategy of catch and slip, let it go a bit, as a way to estimate the coefficient of friction, which would then be used to for proper grasp of objects. But if you think about it, when we as we hold objects or manipulate objects, the number of things that change that contributes towards grip. For example, when you sweat, that alone affects the frictional conditions, but we are able to adjust to that, and so those strategies cannot work. And so that sort of motivates the goal of our project, which is we want to understand how touch receptors in the fingertips signal properties about the objects in the hand, such as manipulative forces and other relevant events that are responsible for dexterous object manipulation. Why do we want to do this? Because this may guide the design of intelligent robots that can perform well in unstructured environments, could be used for haptic devices for remote sensing, and possibly come up with better prosthesis that could be interfaced with individuals that have the amputees, for example, and also possibly come up with strategies for rehabilitation for people that have compromised sensory systems. So the aims of this particular project, what I'm going to focus on for my PhD essentially is we're interested in building an encoding model. That is, we have spike data and we know we have a known stimulus. We want to sort of do a mapping between what we observe from the recordings and what the known stimulus that we apply. We're also interested in building the coding model. That is, if we're only observing spike data, how could we predict what the stimulus that caused that spike response? Now, one of the challenges that that we have is that the technique we're using to record the tactile afferents is limited in how much data we can get, because we're recording single units, and it takes a while to be able to identify a particular response at a particular fiber, and so that takes time, and that means that we have limited data. So the decoding model will require a bit more data than we actually have, and so that brings up the other objective that I want to do, is to be able to implement methods where I can simulate data that has properties that are similar to what we've been able to record and observe. So a bit of background on mechanoreceptors. There's approximately 17,000 touch sensors in the glabrous skin of the human hand. Most of those are concentrated at the fingertips, and those are the most sensitive areas when we are manipulating objects. There are 4 types of tactile afferents. we mostly studied the tactile afferents because we have limited information about the receptors that are actually doing the transduction of what is happening on the outside world and translating into neural code. So 4 of those identified mechanoreceptors we have classified as FA1, the fast adapting type 1, and that is associated with mesinacopaschos. There is the slowly adapting type 1, which is associated with macrodiscs, the fingertips, and then fast adapting type 2, which is associated with Pachynycopaschos, and then slowly adapting type 2 that is associated with Ruffini endings. Now the categorization, the type 1 and type 2 essentially refers to the receptive field. So type 1 meaning they have smaller receptive fields, about 4 millimeter squared area. So and what that means essentially that if we identify an afferent, the spiking, that's the sort of, you can think of as a circular area where you can get response, and outside those borders you basically don't get much of a response. So that's the type 1. The type 2 are sort of larger, they cover a wider area in comparison to the type 1s, and those so they sort of have a single epicenter where you have maximal response, and then away from that you get slowly decreasing response until you eventually get out of within that border. You won't be able to get a spike response, no matter how you simulate it. And basically this just shows how they respond. This is a prototypical characterization of these receptors. You apply a ramp force stimulus, and usually what happens is that the fast adapting types respond to the dynamic aspect of the stimulus, or skin deformation, if you will, and then during the static phase, you see that they don't respond. So they only respond when there's dynamic response. And the slowly adapting types, both types 1 and type 2, do respond to both the dynamic and the static aspects of the stimulus. And here's a bit more properties about the afferent responses. So, for example, the fast adapting type 1s, we know they only respond to dynamic skin deformation, but within that frequency ranges of 5 to 50 hertz. Outside those frequencies, you won't be able to get a response. And they make about 43% of the total mechanoreceptors in the fingertips. Slowly adapting type, they respond to both dynamic and static force, but they respond to really low frequency stimuli or skin deformations. And fast adapting type 2 are highly sensitive to mechanical transients, and actually in experiments you could even, if you blow towards the fingertip, they will respond. If you tap the table, for example, from a distance, you'll still be able to get a response from them. And the slow adapting type 2 that also respond both dynamic and static force. The type 2 receptors are deeper in the skin, whereas the type 1s are more in the dermis and epidermis of the skin tissue. Okay. Now coming to methodology, how we want to approach. So the idea is we want to be able to record signals that are transduced by the mechanoreceptors, and we want to do some analysis so that they will enable us to design models. So we're particularly interested in building an encoding model. So essentially we have a stimulus, we have a spike response. How do we describe that? And then in this case, based on the model that we've used for encoding, we want to use parameters based off that model to simulate tactile afferents that should have similar, at least statistical properties as what we've observed. And then once we have a larger population, then we can perform the inverse problem where we say, okay, these are sort of observed spikes, and from that we want to be able to estimate what the stimulus was. And the other objective, which I sort of started out with, for example, typically most of the models have focused on modeling skin tissues, but we don't know much what is happening within the tissue, or how the forces or how strong the forces are within the tissues. And so moving on from that, our approach would be, if we develop a model, an encoding model, we want to be able to use that to sort of a statistical, because we're going to develop a statistical model for the most part. We want to be able to interface with a neural model, and for example, try to estimate what the mechanical currents that that may cause the response that we observe. So methodology, we use the technique of microneurography. Essentially, we insert electrodes into the median nerve at the wrist. The approach is, we sort of more or less know where they are located. We insert needles, introduce a current pulse, and assess whether an individual is able to have a feeling, so we know that we are actually in the nerve. And then once we have that objective achieved, then we check which of the fingers, because we don't know, it's kind of blind going in, where we can get a response based on which we check essentially each finger and find where we get a response. And if maybe the nerve where the electrode is stuck is corresponding to this finger, then as you touch, you'll be able to get a spike response. And then once that is done, then we go through the classification of what type of afferent is it, and using the prototypical test. And then once that is identified, and then we obviously we program a robot to descend to that particular fingertip and apply the stimulus, and then we'll be able to record.
Why do you have two needles? Do you have two channels, or is it?
No, it's just it's by differential, if you will. I'll just play you a bit of a video here that shows what's happening. So essentially, that we've identified one, and using a von Frey very light fiber to sort of mark the borders of the receptive field, to see how let's say we think this is a type 1, so we want to check and see the area just to make sure that that's what it is, because let's say solar adapting fiber behave similarly, but what distinguishes them is the receptive field, right? So if it responds over a wider area, then we know it's going to be most likely a type 2. Another test that maybe. So that's just to make sure which one it is and sort of map where the receptive field, and sort of that gives us an idea, okay, this is where the receptive field, and it's in an area where you can reach with the robotic arm. Then that we position so that the robotic arm pretty much targets that particular area. So just jump a bit ahead, show with the robotic arm. So we program the robotic arm, and it descends, and upon touching the finger, switches to force control mode, and then applies the force that we selected, for example 4 newtons. And then as we apply the force, then we record. We do touch, just contact, and then we also do slip, because when a graph object, of course, there's some kind of slip, so we want to be able to get some of that information and be able to use it in some way. And back here, so essentially that's what it is, and then we from there we get the data that we have for further analysis. So the first part, once we have the data, is essential. Obviously, the idea of the technique of microneurography is to target a single unit, but in reality we do record other aspects of it. And I forgot to mention that we also use a speaker to try and make sure that we don't have interference from EMGs. So that's how we kind of ensure that we mostly just recording from the nerve. So this is just to do spike sorting essentially. So we may have other signals that we just want to identify occurrences that correspond to a particular nerve fiber that we're recording from. And so this process essentially is we're using the wave shape to identify the particular afferent of interest. But there's also times where there's other, let's say two waveforms happen almost simultaneously. For example, here, that we may not be able to easily recognize the shape of the afferent we're interested in. So here we must do manual editing and look at other waveforms that may have combined to create this sort of complex waveform, and still be able to identify the two individual units that are contributing to this complex waveform. And so, because this is something we have to do, because the technique that we're going to use for modeling assumes that this is sort of perfect spikes that we're recording based on the data. Okay, so modeling we are working within the framework of point processes. Point process being a stochastic process that's composed of binary events that occur in continuous time. So in continuous time, meaning we have an underlying voltage, membrane voltage, but our only observation of it, we assume like it's a spike where maybe after crossing a threshold. And so the model, we're looking at a probabilistic model, essentially, which, but because, so, and the other way to say, well, the point process is to think of it as a generalized Poisson process. A Poisson process is an independent, identically distributed, but we're saying with neural data we have dependencies. For example, if a spike has just happened due to, let's say, refractoriness, you're not going to get a spike. So the spike following after spike is basically dependent on when the spike happens. So there's going to be some latency in that. So there's a bit of dependency in that. And so we use a generalized form of a Poisson process, essentially. But how do we do this in reality? We bin the data, essentially. So we have basically a binary data set. And so just to mention, I forgot to mention here, this sort of history, but this is a combination of the stimulus that caused the spiking activity and any spike history that may contribute towards the probability of firing at a given time instant. So we identify what, like half the force. For example, as I'll show you later, we see that with the solar adapting type, you can see that we apply a stimulus force, but it looks like you see the adaptation that we see initially is sort of influenced by the rate of change of the force. And so we want to have those other things that we can incorporate that we think that are contributing towards the behavior of the neurospike that we observe. So we model this within, we implement our model with using the generalized linear models. Just to go back a little bit, a linear model, we make an assumption that the residuals are normally distributed, and that the mean is linearly is a linear combination of the covariance and the coefficients that we're trying to estimate. But obviously with spike data that's not the case. So we have to sort of use a different type of model to fit our regression. So we work within the general framework to be able to estimate our parameters. That is the first thing that the GLA model does, is to generalize and say, okay, we're looking more into an exponential family of distributions, which will include a number of them, like Poisson, Bernoulli, etc., etc. So, and then one of the things we would identify is a link function, which is used to model the mean, because we are saying we don't have the mean is not a linear combination of whatever the covariates are and the parameters we're trying to estimate, but it's transformed by some link function. And for the case of the Poisson, if that's our distribution, then if we put this into the form of the exponential family of distribution, this is what we have essentially, and this sort of identifies our link function that we're going to use, which will influence how we're going to model our data. And so basically the mean is described as follows. And the other methodologies we're using within the Then the point process framework is the time rescaling, which is essentially a way to transform point processes that, as I said, like the spike data is dependent. So we want to, once we fit a model, we do transform the data into a Poisson process within rate. So that's what the time rescaling does. And the reason we're doing that is obviously if knowing that the data we have is dependent, we would have to come up with, if we want to assess our models with complicated methods to be able to assess our models. But if we do using this time rescaling approach, if we transform it to Poisson rate within rate, which is IID, then we can use statistical tests that are already available to assess how well the model fits the data. And so that's one thing. And we're also using this because we're interested in simulating data. So we're using the same technique, time rescaling, to be able to simulate data that has, for example, that will be able to give us properties that are similar to the sort of data we observe in combination with other covariates, essentially. So this is just using the time rescaling to simulate the spike data. So essentially, well, you know, we sort of break it down into, we're saying that we have the stimulus has an effect on the spiking activity, so obviously it's contributing, but the spike history, which will look into physiological factors like refractoriness, also comes in and modulates so no matter how high the stimulus may be, for as long as the spike has just happened, the spike history will sort of decrease the probability of the spike happening immediately after, until after a certain period of time. And then, yeah, so here we're basically estimating, you know, we simulate an exponentially distributed random variable with mean 1, and then, so we start off, let's say, from 0, and then the goal is to estimate that, given what the rate is, to estimate that, and that will be the spike time as we go along. So we do this continuously until we get a set of spikes. And then the other way is, okay, so this is the model to fit to the data. So we break it down into two components, as just shown you. So there's the stimulus aspect of effect of the probability of spiking, and then we also have the here we're only considering the spike history effect on spiking. Of course, there may be other influences like, you know, the network or the neighboring afferents, but we don't have much of that information. And the technique we use in recording, we're recording only a single fiber at a time, so we don't have additional information. But saying, well, but if we incorporate spiking history, that would lead to a better model that can be used. Here, this is the model I used for, I did a simulation of data, and the goal was to show that we can fit a model, and then show that we would have an improved model if we consider the history effect of spiking, essentially. So in this case, of course, I simulated. Okay, I'll just show you here quickly. So this is an example of what is recorded from actual data. This is a slow adapting type 1 fiber. I took as an example. And so what I did then was look at the distribution of the interspike interval. So based on that, I said, okay, we can select a set of parameters that are appropriate to give us simulated data that has similar properties, at least this sort of distribution. So I generated a simulated form of the force stimulus, took its derivative. Based on what we see, we think that the derivative is having an effect to this adaptation that we see here. Combine it with the force and use this as the sort of stimulus effect to what the spiking is, and then selected history parameters that will govern how fast it will fire immediately after. So in this case, I'm saying like, well, I don't see any firing once the spike has fired, then I have to wait about another 14 milliseconds before I see another spike. So and that's what I said as the sort of I wouldn't say refractory, but yeah, sort of refractory in that sense, in that, well, I have to wait about that amount for this particular. Let's go over this again, just so you can see. So what I was saying is that here's an example of a recorded signal. This is the force that was applied, and this is the spike response that was observed. And what I did is that I took the interspike intervals and sort of plotted their distribution. And this was to give me an idea of how I should sort of simulate spike data that has similar properties to this. And the property I'm using essentially is the distribution in this case. So just going back to the model, I'm saying that, well, I did take a combination of the derivative of the force that I simulated and the force to contribute towards the generation of the spike train. But instead of putting that directly into the model, I'm just saying I'm just modeling this using orthogonal pulse functions, essentially. And so I'm just sort of assuming that, well, I don't know exactly what the stimulus was, but can I still be able to estimate reasonably well what, can I develop a model that can estimate reasonably well towards that? And this, you can think of this as, this is sort of equivalent to a post-stimulus histogram. And then this is the model, the part that models the spike history. So we estimate parameters and then fit the model. Okay, so fit the model, used GLM fit in MATLAB to estimate my parameters, and then of course go back to, yeah, of course this is okay, the technique I'm using to estimate what the mean is, or the rate is. And because I have two models, eventually once I do, I multiply them together once because I sort of I get coefficients related to this and coefficients related to that, and then I compute the rate and then multiply the rate together eventually to get the final rate. So so I assess how well the model does, essentially. So I did a model first where I didn't consider the history aspect of spiking, which then I fit on the data, and the blue line shows how well the model is. So essentially, okay, so time rescaling, we do time rescaling, we get a Poisson process with unit rate 1, transform that into exponential random variables, and then transform that further into uniformly distributed data, essentially. And so that's what I'm plotting against this. And this is the Kolmogorov-Smirnov test, and the black line is the 45-degree line, and the closer with the 45-degree line, the better your model does. And the dashed lines are the 95% intervals. So then I fitted the model again, considering estimating parameters that relate to history, and then that model, you can see it fits. You know, we still need to do better, but it is more or less, it does a better job. It's a better model compared to the previous model. Yep. And so in summary, essentially, the idea is we're trying to see whether we can develop models that can be sort of reasonable within the point force framework. And that seems to be the case. However, there are still challenges. One of the things that I'll be working on is essentially looking at the nature of the data that we get. For example, with the fast adapting types, we have a period where they only respond to dynamic aspect of the stimulus, and then during static phase you don't get a response, and then, you know. So the idea is to try and do the parameter estimation in a dynamic way. So we're trying to develop a model that state space model that using Bayesian techniques to essentially we're saying that the parameters are not going to be constant across as you go, so they're going to change as the stimulus changes irrespective. And so that's one thing that we'll be working on to try and see. The other aspect, what I've done is I fitted this on simulated data. I know what the history is, and so it was easy to know what to do. What I still have to work out is I want to be able to get data. I want to be able to work out, what the history window is given my data that I have. And that, I think when I do through wherever we were fortunate, we got some data that was spontaneously spiking, and my idea is to use that spontaneous spiking data to sort of estimate how a spike, how history affects the spiking at a current time, because that I'm saying, well, I don't have a stimulus in that, and it is spiking. Okay, I don't know exactly what is causing that, but I'll make that assumption that, well, it's sort of self-driven in a sense, and then I use that to see how well that to assess how far back in history do you have an effect on spiking, and then use that as my history window. And then the other objective is to be able to predict the force stimulus given just the spike data. Once we fit a proper model onto the data and be able to simulate data, and then that should be something we'll be able to do. I just want to show you an example of the spontaneous spiking data here, just to. So this was just, okay, let me just, this was just a backup slide. This just we're trying to see how, for example, this is the rate, the firing rate, and this is the derivative of the force. And so we're basically just trying to assess, see how, how the stimulus is affecting the firing rate of some of these cells. Let's just, and so this is an example I was showing that we had a case where we have it's spontaneous spiking, and then we apply a stimulus. It sort of gets into the fast adapting type behavior. It modulates a bit, and then during the plateau it cuts off, and then it goes back to that. So what I'm proposing is that we can use some of this data to give us an assessment of how well or how the spike history affects spiking at the time. And with that, I welcome comments, questions, suggestions, and thank you for listening. You mentioned about interfacing with the neural model. Right. So that's the last sort of step. Yeah, so that will be more after we develop models. We know we can, we have dynamic models that can estimate parameters online, and then that would be the time to think about how to interface it exactly with. I have a colleague of mine is actually working directly with that, but so we sort of have complementary projects. So I guess whatever I find or whatever I do will be useful for his work as well, because he's really more interested in actually interfacing with interfacing robotic prosthesis with amputees. Yeah.
How much do you have to factor age and any other biological responses in your model?
So we're collecting this data from young individuals, healthy young individuals between the ages of 18 and 40. So we wanted to, as a start, to keep it to that, so that we believe that these are more people that have better responses. And so
how would you think it would change over the course of time then, or over the course of age?
Of course, sensation does degrade over time, but I don't know exactly how that. How to quantify that at the moment. So that's why we're mostly focusing on people we think that are healthy and still, because we're still at the stage of understanding what to do. And then I guess once we have that, then the next stage will be to look into those differences. Yeah.
Patrick, thanks for the talk.
Yep.
I just had a question about using this in the development of, let's say, robotic arms, for example.
All right.
What type of sensor do you think we'll need to actually get the information about the sensation, about the tactile?
Right. So there's, we think of, let's say, can we design sensors that. So I think one of the main things that we're trying to understand is the mechanotransduction aspect of it, and that's still a bit of a challenge because of the anatomy. For example, I was talking about the macro discs. There's a bit of information now about understanding of how it behaves, but still we know that an afferent fiber is connecting to several of those, right? So if you want to model that, so how do you, we don't know precisely how many contribute, how they contribute, or how they worked together as a whole to connect to that particular afferent. So that's still a bit of a challenge. So yeah, mechanotransduction is something that's of interest, but at the present moment it's still a challenge. People have tried that, but they, like I was talking about, they're trying to model skin stress, but it's still also it's sort of arbitrary, really, because they come up with a method where I say, well, this is the skin stress, and we can convert this to current, and then we drive a neural model. But that current is sort of arbitrary because you kind of pick any number and make it work, pretty much, depending on how you design it. So yeah, so it's still, but I think that as information becomes available about the receptors themselves, this will provide ideas of how to sort of go about that and see how we're going to be able to take an external force and transduce into neural code that's sort of similar to how the real receptors are doing. And also it'd be interesting how does that, because obviously when we stimulate the fingertip, we're stimulating a number of different types, all the types, and they must be working somehow in sync together to give pro information. How is that information integrated, and where is it integrated to? All those are questions we don't know at this point. Yeah.
More on the modelling side, as far as specifically what you've been doing. I was just wondering about how data intensive this approach is if you were wanting to do this live, so real time, for example. How much of a history window, so buffer, would you need? How intensive is this data analysis process?
So obviously, so one of the things that I thought about when doing this is the decoding, especially. I was thinking of using it. Like obviously, if you use particle filters, they're great. We don't need to worry about anything. But obviously, they're going to be data intensive. So my focus at the moment is try and develop sort of a Kalman filter that is, tailored for spike data, because in that case, at least that will alleviate on the. It will perform faster compared to, we don't need as many computational resources as we do for the other. So it's a compromise, really. So yeah. Yeah. Thanks. Yeah. Yes.
I know that there is a group of Silvestro Mussera in Pisa
Right.
in 88.
Yeah.
And they have developed this fantastic
robots.
bio-hand.
Right. Yeah.
So robotic hand, and which has very nice precision as well, and which people can actually move using EMG signal.
Right.
And do you think that, how does it feel with your work? Because with your work, maybe that would be like a sort of real hand with those possibilities of sensation,
yeah.
and not only obviously mechanical movement like.
Yeah, so I guess that's the challenge, because theirs is EMG based, and it obviously has limitations. I've seen there was a video they put up together where they were training someone to recognize shapes, but he has to train, and I think, but it involves retraining and retraining in order for it to work well. Actually, Sylvester at the conference and just like, well, an ugly solution is better than no solution. Yes, it is a solution at the moment, but I think it would be good to sort of try and find other solutions that would provide better solutions. If we, yeah. Thank you.